Module 5, the muscular system, histology and physiology. How a muscle fiber contracts. Well, this is the section where you will learn more specifically about one of the main characteristics of muscle tissue. It has contractility. That means it's able to contract, and that produces movement. Now that you've learned about the structures of a muscle, let's take a look at what's going on at the cellular level when a muscle contracts. For a muscle to contract, the muscle fibers need to shorten. The myofibrils are the mechanisms for this to happen. This process is described as the sliding filament model, and it helps you to see not only how muscle fibers are able to shorten, but also how the entire muscle contracts. Remember in the last section how I said that the myofibrils are arranged into repeating units? That gives the muscle fibers striations, or their stripiness. We call these repeating units sarcomeres. A sarcomere is a repeating unit of a myofibril. Sarcomeres are connected one end to another, and they're what form the myofibril. Take a look at this simplified image of a sarcomere. Because sarcomeres are lined up end to end to create the myofibril, when you see what happens in one sarcomere, you will understand what happens to the myofibril, the muscle fiber, and the entire muscle too. Muscle is made up of primarily proteins. In this sarcomere, you can easily see two of them. Myosin forms the myosin myofilament. Now, you've heard lots of new vocabulary which uses the prefix myo. Myo means muscle. The end of that word sin refers to protein, so myosin means muscle protein. Of the two major proteins in a sarcomere, the myosin myofilament is thicker. The other protein is called actin, and it's the thinner of the two. Skeletal muscles show striations because of the thicker myosin myofilaments and the thinner actin myofilaments. When you viewed the skeletal muscle tissue under the microscope in the previous lab, you could actually see the myosin and actin myofilaments within the sarcomere. The thick dark lines you saw were the thicker myosin, and the lighter stripes were the actin. We call the thick stripes A bands, and the lighter areas I bands. So in the diagram, this is an A band, and these are I bands. The extensions of the myosin myofilament, called heads, attach to the actin myofilament at active sites. These joints are called cross bridges, and these are the foundation of muscle contraction. What happens is the myosin head grabs the actin at the active site and pulls it to shorten the length of the sarcomere. When all the myosin heads of all the sarcomeres do this, the entire muscle filament, and thus the entire muscle fiber, contracts and shortens. The myosin heads all work together to pull at the same time, creating a strong force of contraction. In the diagram, you can see a vertical zigzag line. That represents one more feature of a sarcomere. These are Z-discs. You can remember this term because of the zigzag pattern they make. Z-discs are also made from protein and are for anchoring the actin myofilaments. And they're also the location of the ends of the sarcomere. The last zone is the H zone, and it's the location inside the A band where there is no actin protein. So, to sum up, a sarcomere is bordered on its edges by Z-discs. They separate one sarcomere from another one. Actin myofilaments connect to the Z-discs, one group on one side of a disc and another group on the other side. The actin myofilaments within a sarcomere are separated in the middle, which is called the H zone. The H zone only has myosin. Myosin myofilaments are thicker than actin and are found inside the actin myofilaments. The myosin heads attach to the active sites of the actin, forming cross bridges. The thick myosin myofilament defines the A band. The parts of the sarcomere where there are actin myofilaments and no myosin myofilaments are where the I bands are. Finally, the sarcomeres are attached to each other end to end to form the myofibril, which is the contractile organelle in the muscle fiber cell. Well, now that you know the basic structure of the sarcomere, I can go over what happens when it contracts. For a sarcomere to contract, the heads of the myosin myofilaments grab onto the actin myofilament and pull in unison like the oars on a boat pulled by the rowers. They all bend back together, pulling the actin myofilaments closer together. When this happens, the Z-discs get pulled closer together and the length of the sarcomere shortens. Well, that means the distance between the actin myofilaments, or the H zone, shortens. Now there's more overlap between the actin myofilament and the myosin myofilament. The sarcomere gets a little thicker. So now you know why your muscle bulges when it contracts. The entire muscle shortens but gets thicker as the actin and myosin myofilaments overlap more. During muscle contraction, the I bands become shorter too. But though the sarcomeres are shorter, you can see how the actual myosin myofilament and the actin myofilaments do not shorten. They've just moved in respect to one another. So that means the length of the A band has not changed because the A band is the length of the myosin. We can say then that during contraction, sarcomeres will contract and shorten, but their myofilaments that do the work do not contract. This is a fascinating process, but it gets even more interesting. Let's take a closer look at the cross bridges where the myosin myofilaments grab onto the active sites of the actin myofilaments. Scientists have learned that the actin myofilament contains two more proteins besides actin. Tropomyosin is a long strand that wraps around the actin molecules, like the stripes on a straw. Troponin lies across the tropomyosin. So let's say you're holding a slice of pizza and you want to eat it. You need to bend your forearm up towards your mouth, so your brain sends a signal across the neurons to the biceps muscle. That signal causes the sarcoplasmic reticulum in the muscle fibers to release calcium ions into the myofibrils. The calcium ions will bind to the troponin, altering its position on the tropomyosin. This makes the tropomyosin roll back along the actin, exposing the active sites on the actin myofilament. The active sites draw the myosin myofilament heads, causing them to bind, and that creates a cross bridge. Basically, what's happening is the neurons cause the sarcoplasmic reticulum to release calcium ions, so the proteins in the active myofilament move around to expose the active sites, which attract the myosin myofilament heads to themselves. The sarcomeres contract, and you can eat your pizza. If you've ever done a large number of sit-ups, you know that contracting your muscles requires energy, doesn't it? Well, remember back to cellular respiration. Cells store energy by taking adenosine diphosphate molecules, or ADP, and binding on a phosphate molecule, P, to form adenosine triphosphate, or ATP. Well, at the end of a muscle's contraction, ATP molecules bind to the myosin myofilament heads and release their stored energy by breaking down into ADP. The released energy gives power to the myosin myofilament heads so they can do their job. You see, it takes energy for the myosin heads to attach to the actin and move. And the only way a cell can provide that energy is by breaking down ATP to ADP. So after the myosin myofilament heads bind to the actin, there's still ADP bound to them as a remnant of its release of energy. And that energy enables the myosin heads to pull the actin myofilament. As they do this, the ADP falls off the heads and gets recycled, along with the phosphate group that was broken off back into ATP during the later cellular respiration. When the sarcomeres contract, it's called a power stroke. But there's more to this process. The ATP had enabled the myosin heads to pull one step towards the center of the sarcomere, and the remaining ADP and phosphate have fallen off. Well, now more ATP will come in and bind to the myosin head, releasing its energy by breaking down into ADP and phosphate, and enable the myosin head to release its bond to the active site of the actin, breaking the cross bridge. All of this occurs to enable the myosin heads to make just one swipe of their oars, or one power stroke. But the myosin heads are now bent backwards, so more ATP comes in, breaks down into ADP and phosphate. The energy moves the myosin heads back so they're ready to flex another time. And this step is called the return stroke. The muscle can continue to contract only as long as the energy is there to keep those myosin heads grabbing the actin active sites and pulling on them. Since there's ATP within the cells, this process will occur whenever there's access to the active sites, and that has to do with the tropomyosin rolling back to expose them. When you want to stop contracting a muscle, you again send signals along your neurons. This signals the sarcoplasmic reticulum to move the calcium ions away from the myofibrils using active transport. With the calcium ions gone, the troponin resumes its original shape, causing the tropomyosin to roll back and hide the active sites on the actin myofilaments. Now the myosin heads cannot connect to them. At this point, the muscle is no longer contracting.